My audience for this project is my 13-year-old sister named Maddie. Hi Mads, I miss you and I love you, and today I'm going to show you this protein that I made for my biochemistry project. This is the protein Kras, and just try to maybe understand. I'll try to simplify it a little bit, but let's get going. So in your body, your cells are constantly telling or deciding when to grow or stop growing, and Kras is like the switch that tells them what to do. So first, it's located on the inner surface of the cell membrane. So here's my cell membrane, and it's going to be on the inner side of it. So we have inner and outer side, if you can see. So it's going to be on the inside of the cell membrane. It sits right along the membrane so it can receive signals from the outside and then tell it to the inside of the cell and pass it inside. Kras is a globular protein, which means it folds into a compact 3D shape. So we got the 3D part of it. It's really important because it tells, like it decides how to interact with other molecules. And is attached to the membrane by a lipid modification that anchors it in place. So we have this little thing that's going to anchor it to the cell membrane. It's going to anchor it in place so it can be that signal transducing. The fatty acid tail, so the real one has a fatty acid tail, but this is just the tape, and it's just connecting it to that membrane to keep it stuck in place. Okay, now we're going to talk about the different structures. So we have primary, secondary, tertiary, and quaternary. So primary is first, just primary, right? Put that down. That's here. We've got this chain of amino acids, kind of looks like beads on a necklace, right? Amino acids are the building blocks of proteins, so we need these amino acids in order for this protein to be a thing. The Kras protein is made of 188, so that's like a lot, but we're just going to show those for now. Then after that amino acid chain forms, part of it folds into a smaller repeating shapes, and this is the secondary. So we're on number 2, secondary structures. So we have the alpha helices here, which are like the spiral shape, and then we have the beta pleats, which are the zigzag right here. Excuse me, beta sheets. And it's more like, the zigzag. So then we've got the spiral here. And the next level is the tertiary structure, which is really just the full 3-dimensional folding of the entire protein. Let me know if you need me to stop and repeat anything. I know I'm going kind of fast, but I like to talk fast, you know this. And so in this model, the ball represents how that chain folds into a really compact structure. And then we have this connecting point here, which these connections here represent interactions between amino acids that help stabilize that shape. So these are interacting constantly to make sure we have that tertiary structure, which is really cool. But yeah. And then some proteins also have a quaternary structure, which is just like a bunch of protein chains combined together to make one big protein complex. But luckily, Kras does not have that because it's made from only one polypeptide chain. So that's really nice. So Kras acts like a molecular switch. So we have GTP right here and GDP on top. The GDP is the blue one. And so when bound to GTP, the green, it's active, and when bound to GDP, it's inactive. So then we have these strings here, which represent the signals that Kras sends to other proteins in the cell. So they're going to tell it to either, it's going to help with cell survival, cell growth, and cell division, if that's the right way. I don't know if it'll be the right way. So that's just, representing the signals that it's sending to other cells. So then we have this red bead, which I'm going to place it right on here, and it's going to act as a mutation. And so it's a mutation in one of the amino acids of the Kras protein. So it happens when the DNA sequence changes, and it causes a different amino acid to be placed in the protein, which we don't want that other amino acid. We want the normal amino acids. We do not want this red bead to be here, but it is here. And because of it, Kras can be stuck on the on position. So instead of going between active and inactive, it's constantly telling the cell to grow and divide, which leads to cancer. We know when it grows super fast and uncontrollably, it's cancer. So mutations in Kras are commonly found in cancers like pancreatic cancer, colorectal cancer, and lung cancer. So because that protein is always stuck on, the cells are dividing uncontrollably. Some limitations you might be able to see here, mods, maybe a little bit. The model's not to scale. Proteins are way, 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 way smaller than this thing is, but I had to show it big so I could get into all the nitty-gritty of it. Also, the folding of the proteins in real life is much more complex than what this model shows. So the different primary, secondary structures are a lot more complex, but I try to make it as simple as possible. And then finally, proteins are constantly moving and changing shape in the cells, but this one's static because I can't necessarily change all the shape of it all the time. So those are my limitations, but I love you, Mads, and I hope you enjoyed learning about Kras. Bye.